This is Ben Newman. Let's try this. So I've got 16 tabs open with 16 different papers. These are all either preprints or things that are published but are not available in the final form but are about to be. So these are things that are coming down the pipeline or are already there. They all have to do with SARS coronavirus 2. And we're going to try and go through these and round them up just as fast as possible. So let's try this. And I don't know, let me know if you like this. Yeah, let me know if the sound of my phone dinging in the background is annoying. Here we go. All right, article number 1, Mdpi, right there's the title. And this is interesting because it's showing that interleukin 6, we already know from several other studies that this is heavily involved in the way that coronavirus causes disease. This seems to be one of the signals that is most raised in people with severe disease. And this looks like it is it's actually derived from a coffee bean. That's why it's called javamide. Yeah, clever name. But javamide 2 does seem to inhibit interleukin 6. And it doesn't infect some of the other things that macrophages would like to be able to respond to, like interleukin 1 beta and TNF alpha. These are just other parts of the immune system. So there's that. Interesting. Yeah. This is from the USDA. It is not... it's done in cell culture. It's not even done in animals yet. So we're a ways from being able to use this in people, but it's promising. Next paper. All right, so this one is about bats, and I'll flick up to the title right at the end of it. But this is looking at the kinds of coronaviruses that bats have and the way that the coronaviruses seem to move between different kinds of bats. So alpha coronaviruses are things like 229E and NL63. These are human coronaviruses that we would call common cold viruses, and oh my goodness, are they prevalent in bats. We've got a bunch of different ones here. We've got your horseshoe bats, we've got your leaf-nosed bats, we've got your vesper bats or evening bats, we've got your long-winged or bent-winged bats. Yeah, you can see that from the pie chart, most of them tend to be within a group. So all the members of a group will have similar viruses, but there's going to be a little wedge in each group where it's a different kind of bat that seems to have this virus. So coronaviruses aren't just spreading from bats to human, they're spreading from bat to bat and different kinds of bats to some extent. When they're there, they are competing with the other viruses that are already in the bats, and so it doesn't look like they're getting a giant foothold. But yeah, the picture is a little cloudy. So that's alpha coronaviruses. We really want to know about beta coronaviruses. These are the ones related to MERS. These are the ones related to SARS. And these are what we would call the Group D and Group E coronaviruses related to HKU9 and Zhejiang 2013. Neither of these has ever infected a person yet. These cause terrible things in people, Group B and Group C. And as you can see, the beta coronaviruses tend to be a little more restricted. It looks like they're not as good at jumping, except for this one group over here where you've got some members of the group, or rather, members of other groups that are infected. These guys in sort of light green here are neat because all the rest of the bats on here are what you would call mini bats or even micro bats. They're little tiny bats. And in terms of bat phylogenetic lineages, that doesn't mean a whole lot. All the tiny ones aren't related to each other, no. Just because they look the same to you doesn't mean they're all related. But these are the big fruit bats. And so fruit bats do have coronavirus, and these are sometimes called megabats over here. They are just not coronaviruses that have currently infected anybody, but this is another thing that is out there and that's worth watching out for. So that paper is, scroll up a little more. There we go. That's the paper. And so you can pause it and go read that if you want. Okay, next one. So this is one that we talked about on the Facebook thing. This is a really nice study looking at where do antibodies that bind to the spike tend to bind, and particularly the ones that neutralize, that can shut down the spike, where do they bind? So the main locations where they bind are, one, right on that receptor binding domain, which is the part of spike protein that reaches up and tries to grab your receptor. And so, yeah, sticking to the end of that and blocking it is just directly blocking the spike from doing its job, and that's fantastic. We love that. Other interesting thing, a little further down, I think it's another in figure 5 or 6, actually figure 6 of this paper, it's actually modeling and showing that, you know what, an antibody has, this is just the tip of the antibody, and there's two of those tips because the antibody is Y-shaped and it has two of them. It's showing that the spikes are positioned just right on the outside of the virus that you could have a antibody and could reach between two of those spikes. Let's see if we can find that. No, maybe it's figure 7. There we go. There we go. And so they're saying, well, yeah, these are the tips of the antibody. This is how they bind. This is how big a full antibody would be. It's tough to get the structure of a full antibody because it's kind of bendy around these parts. And so we know what the ends are going to look like, about how far apart they are, but this is all very twisty. They're like little swivel hinges in there. Yeah, this may be in part why some of these antibodies are so good at shutting down the spike, is because each antibody is not shutting down just one spike, it's shutting down 2 at a time. And yeah, that's the way we want to take down coronavirus, 2 at a time. Love it. All right, what's next? Okay, so from, oh, sorry, we should go back. This paper is called Structures of blah, blah, blah. There we go. Next paper. All right, so this one is another one of these big, let's look at the transcriptional profile, what genes are being made inside of every kind of cell and just everywhere. Yeah. And these are super expensive studies, very interesting, sometimes difficult to figure out what to do with the information once you have it. And they tend to blur some things, like you'll take a bunch of cells from a tissue sometimes, in some of these studies, not this particular one, and you won't know if they're, you can say that, these two things are up in this tissue, but you won't know if they're up on the same cell or up on like an adjacent cell, something like that. Anyway, what they find in this paper is that, no surprise, most of the damage in COVID-19 comes from these, what they're calling myeloid cells, which is your macrophages, and all of your granulocytes, your basophils and neutrophils and eosinophils and just all of those guys. And yeah, together they make up pretty much all of the innate immune system, and that is exactly what we've known all the way along. But the interesting thing that they find is that, yeah, it's some of these cells, but it's different populations of the cells that are causing different problems in different places. And so I think that's interesting, and this is probably a springboard for more different kinds of research, or maybe this is summing up a bunch of other reports where they just look at individual parts of the body or individual cell types. But yeah, it's nice to see a little holistic approach to this thing, and so there's our paper. Next one, this is a real quick one. So this is looking at the E protein of the coronavirus, and the E protein is probably most involved in releasing the virus from the cell, but also under some conditions it can be an ion channel, so it can let ions from outside to inside the cell, and maybe that can affect the way some of the other proteins stick together. So this might be a protein, or rather this protein similar to this in other viruses, help the virus get ready to enter a cell by sort of breaking up the structure inside the virion to get a little looser so it's ready to dump out its contents into the cell. This is looking which end is up in the E protein, which, weird though it seems, has been a subject of some controversy in the past. And they're looking for which parts of this they can actually tag with, I believe it's a glycosylation tag. And it turns out that, yeah, it's oriented like this. So the beginning of the protein called the N-terminus is going to be on the outside, and the end of the protein called the C-terminus is going to be on the inside. And in addition to this, we think this probably spirals up into a little helix, and half of the helix is probably going to be buried in the membrane. Those are the best predictions that we have right now. So there we go. And E's are going to club up. There are going to be 5 of them together at a time. And so that's that paper. Yeah, neat. All right, next paper. Oh, and did we show that paper? No, we got to show that paper. There we go. That's the name of the paper. Next paper. So this one is looking at, so the rheumatoid synovium, I think. Isn't that right? Yes, okay. So this is on your joints. This is like a little wrapper layer. And what they're showing is that when you have a lot of interleukin-6 around, so figure 1D hits that nicely, so interleukin-6 is plus, then you will have lots of ACE2, which is the virus receptor. So this is trying to show why, if you had a situation where there was interleukin-6, you may end up potentially leading to a situation where joints could become infected. Now, this is another one of those things to file away under could happen, but there's no evidence that it does. But interesting, I guess it also shows that potentially, and they didn't look in this paper, but potentially if you have rheumatoid arthritis already and high interleukin-6 levels, maybe that could lead to more severe disease. And again, it's something we don't know, but worth keeping an eye on, and interesting for that. The next study is, yes, part heartbreaking and part really good science. So they've got this figure 2 in here, which I love. They're showing what they were doing. They're like, we're making so much progress. So we got the protein to express on the 9th of February. We got our first crystals of the proteins. They're trying to crystallize the protease, the main protease of coronavirus, in order to figure out what does it look like and what drugs bind it, can we slow it down, because this is a very important protein to the virus, and it's something that if we can take it away from the virus, then the virus, essentially is no longer a virus. And they're showing that, we had lots of crystals, lots. We've done so many samples, thousands of samples. And then just at the point where we started to get some data back, the beamline. So this is how you do your X-ray crystallography. You've got to go to a beamline. And if the beamline at your facility gets shut down, then bam, you don't get to do any more structural studies. And so they took those lemons and made some really tasty lemonade out of them. And then over here is the heartbreaking part. They're showing other people's structures released. And so here's the first bunch of structures that come in early March. And boy, it looks like they could have got there. Maybe it would have been close, but they would have been among the first ones. And so what they're doing now is this study where they take all these crystals that they've got. So they've got tons of crystals. They just can't solve the structure of them because their beamline is shut down. But you can do some things. They probably have a little in-house X-ray source, like a little teeny-weeny one that can tell them some stuff but isn't good enough for big structural data sets that they need. And so they just started soaking the crystals with different compounds and seeing where those compounds stick, because you can tell that on a little tiny home source. And so, I think the nice, there you go. It's summed up very well in the figure 3 legend. Bound fragments, so all the little things they're putting in, all the little drugs and stuff, sample the active site, which is the part of the protein that actually does this, comprehensively, which means that pretty much everything they put in that had some effect on the protein actually goes in and actually just sticks to this area called the active site, and bam, so they identified something like 70 new compounds that can block the active site of the main protease, at least to some extent. Doesn't mean these compounds are not going to have side effects, because there's a lot of different protein cutting enzymes in the cell, and some of them are actually somewhat similar in the way that they work and the structure to the coronavirus enzyme. So, but a really neat study where they managed to get something out of almost nothing, out of probably great frustration. All right, sorry, we should show you who did that and where. These folks and there. Okay, next one. So this is looking at a mouse model, and this is what they're basically doing is hot wiring a mouse. So right now there are these human receptor-containing mice, human ACE2 transgenic mice, or what they're called. But they only exist on one genetic background. So you can't choose from a variety of different mice that have slightly different responses and triage your virus in any of those. You've got to use the one that's available. And it doesn't even matter, because even though that one is available, all of those mice have been spoken for a while now. And so if you want them, you can't get them. Yeah, too bad. So what's a person to do? Well, like I said, they're going to hotwire the mouse. They're going to take a thing called adeno-associated virus, which is a little thing that's normally associated with a virus called adenovirus that causes, I don't know, it can make your tonsils swell up. It can cause something like a cold, but yeah, it's a pretty common thing that people get. And then adeno-associated virus is like a virus of that virus. It's associated with it. Its cool property is it'll go into a cell and it'll drop off its gene, and it'll actually cut the DNA of the cell and glue its gene into that spot. And it's fairly careful about how it does this, not 100% careful like I've heard some people claim, but pretty careful. So, all right, that's good. And so what they're doing is they're injecting this adeno-associated virus with human ACE2 inside of it. And they're using that, they're putting that into the lungs of the mice, letting that just deliver human ACE2. And so bam, you get effectively a human ACE2 mouse where the human ACE2 is only expressed in the lung, which means you can then infect these mice. And so they're infecting them, and sure enough, it's infectable, and they reproduce some of the symptoms of COVID in this one. This is a neat thing. So the problem with the other mouse model, the human ACE2 transgenic mouse model, like done the regular way, not the hot-wired way, is that they have ACE2 expression in lots of different tissues. And you put in the virus, doesn't really matter how you put in the virus, but you put in the virus and it goes straight to the brain, and they die of inflammation in the brain, encephalitis. Yeah, which is not how people normally die from SARS-CoV. But when the receptor is expressed in more places, then you get a broader infection. And so, yeah, it's an interesting model, but not, a super great model. And this crummy, cobbled together model is actually, I don't know, might actually be better in some ways. So here are the people that did that one. So that's kind of neat. All right, next one over here. So we still don't know what TMPRSS2 looks like. And you remember, this is one of the important proteins. This is going to reach around and cut the coronavirus. The blue region up here is what cuts it. And so what they've done is taken a lot of similar proteases and they've done a model of it. And so that's a model, and that's probably going to be pretty close to the truth. But until somebody actually solves the structure, we're not going to know how close to the truth they got. And I think they're just modeling the green and blue parts, and they're imagining you're going to have to cut off this part, which is probably going to be the case. And so, I don't know, hopefully we'll see a real structure of that soon. But the best part about this paper is they look at human variants. So there are a lot of people walking around out there with different changes to TMPRSS2. And they're looking at these things and running predictions based on what we know about similar proteins. What would these mutations do? Well, it turns out most of these mutations, one of which is very common. It's there in about a quarter of the population, with 6+% of people actually only having this variant, so that's kind of neat. It's predicted to be damaging in the sense that TMPRSS2 won't work. By a couple of prediction programs and some other stuff that they talk about here. The variant is a valine, V at one amino acid 160, and it's turning into a methionine, which is just a much bigger amino acid. And so, valine 160 to methionine, what they're saying here is that if you've got people that have this gene, their TMPRSS2 probably isn't going to work as well, and somebody ought to really look and check through the DNA of people who have had this and had severe or mild COVID, and just have a look and see if this is helping. Because in theory, having a TMPRSS2 that doesn't work very well should, it can potentially hurt you in some way, but it should hurt the virus a whole lot. This seems to be a really important way that the virus gets in. So, interesting. Maybe that's going to be a thing. I don't know. Let's find out. And boy, I do this every time. Boom, there's our paper, there's our people, our preprint rather. Yeah, maybe someday it'll be a real boy, who knows. Okay, intestinal inflammation modulates the expression of ACE2 and TMPRSS2 and potentially overlaps with the pathogenesis, so fine. This is a paper saying that when you get inflammation in your intestine, so think irritable bowel disease, or think SARS-CoV-2 expression causing an immune response in your intestines, similar to what happens in the lungs, but carried out by slightly different populations of cells. That's actually going to push up the amount of ACE2 and TMPRSS2, so more inflammation, at least in the intestine, leads to more receptor being made, which potentially leads to more virus getting in, and so you have this feedback cycle to think about as well. This may be why intestinal infection is so common in SARS coronavirus 2, and it certainly seems to be. So interesting. There we go. All right, next one. So this is looking at the effect of age on expression of TMPRSS2. So we already know that ACE2 does seem to go up to some extent with age, although in this paper, and they're looking in mice, the effect is not as strong with ACE2, but boy, is it a lot stronger with TMPRSS2. So this is the one right down here. Well, I can't quite point at it, but there. So look right where I'm circling, and then, all right, let's back off there. So what we've got is on the blue side, we've got mice at different stages. So these are stages of development before birth, way before birth is the little one, and you can see there's not much TMPRSS2. Like the higher those blobs go and the more stuff is up high, it means there's more TMPRSS2 expression going on. And you can see that we go from the blue at the one end, so this is way pre-birth, to the adult stage at the other end, P64, 64 days after birth, I think. And you can see that the amount of TMPRSS2 has gone up massively. And so they go on to show that this is probably not just mice, this probably also happens in humans. And so TMPRSS2, this activating protease, may be an important thing that gets the virus in. And yeah, that's a neat little thing. So click out of this, go back there. That's our paper. That's where this is coming from. All right, next one is, shoot, I forget what this paper is. Let's see. Okay, fine. So this is looking at what antibodies you're making and where you're making them during infection. Yeah, and this was a very sad graph. That's why I pulled it up. Yeah, there we go. All right, so let's pull it up again. So on this side, you got your mild cases. On this side, you got your severe cases. And they're also doing this in young and old patients. I think B is young and C is old. And what they're showing is, yeah, kind of sad all the way around. So mild cases, this is the amount of antibody that you make. And just look at those wimpy little curves. They go up, and then the IgA curve comes down. After about a month after you start to get symptoms, you run out of IgA, which is probably going to be the most protective antibody that'll stop you from getting infected again. You're basically back down to where you were before you had symptoms in the first place. So that's terrible. Mild cases are most likely going to be reinfectable at some point. May take longer than a month or two, but yeah, probably this is a thing that is going to happen. Severe cases, though, seem to make much better antibody responses. I wouldn't recommend going out and getting a severe case because that's where the death tends to come in as well. But yeah, it's a better way. Being young and having severe COVID does seem to give you good, better, higher antibody responses. Then scroll down to the bottom. So what about being old? Well, yeah, being old's not great. And here we go. So you just got lower responses to the non-severe, and you've got a real mixed response, even if you've had moderate to severe. Look at all those cases down toward the bottom. So I'm just kind of pointing toward them because if I get too close, the figure legend's going to come up and block them out. You got a few people up here at the top that are making real good responses. So the higher up the dot is, the better the response is. But look at those people down at the bottom. Oh my gosh, there's so many of them there. And so this means older people, even if they've had severe cases, some percentage of them are probably going to be reinfectable, and it's probably going to be sooner rather than later. You're not going to know. Just because you've had a severe case, you will not be able to tell that whether you are protected. This is just reading out the antibody levels without even going into the rest of that. So it would be good to have kind of a test that can read out antibody levels and tell you which ones are actually protective and which ones would give you a better chance of withstanding it. So there we go. Here's our paper. Bam, right there. Yeah, interesting. Interesting preprint. Yeah. Sad and bad for the future of the world, or maybe just a wake-up call, like anybody's already not had enough of those, but that we need to do something about this, or else it's going to continue to be bad, and maybe even increasingly bad forever. Okay, this next one doesn't even, you don't even have to go through the graphs, because the graphs are just all how can we destroy SARS-CoV-2 in a way that lets us do downstream assays. And so you can cook it at 80 degrees Celsius, yeah. And so that's not even boiling, but that does seem to destroy the virus very effectively. A range of detergents, so things like soaps, but also things you would use in the lab, those quaternary ammonium detergents, things like that. And they look at ultraviolet, so we've heard about UVC, ultraviolet type C, things being pretty good at knocking out the virus. And sure enough, this is a nice paper that goes through all of those different ways and just provides you with a variety of methods to destroy SARS coronavirus 2. They're talking about for people to use in labs in downstream assays, but these are all methods that would destroy SARS-CoV-2 inside or outside of the lab. Yeah, so this is good for people to know. All right, next one. So this is talking about a protein that nobody knew existed before, and it's this little yellow guy right here. So this is SARS coronavirus, or rather these are coronavirus genomes. And so they're shown here, the spike gene is right there, and so you can see it's in the same spot in all of them. And then E and M are always right together, one right in front of the other. And so they're right about here. The nucleoprotein is N is here. These are the four that are conserved, always, always, always. And then there are going to be in viruses related to SARS coronaviruses. So these are in the subgenus called sarbecovirus. And these are all going to be genes that are called accessories. These are going to help out the virus in some way or hurt the host in some way. And they exist in some strains, not in others. And yeah. Some of these have been talked about before. Some of these are kind of overlapping other genes. So just like a different way you can read the same piece of RNA and make a different little protein out of it. And two of these strains only, the pangolin and the SARS coronavirus 2 from people, have a 3C in addition to the 3A and the 3B. And I guess we should have renamed this one 3B, but we'd already found a 3B and it's the one down here. So they're keeping the naming consistent and I respect that. So they're saying this is a new thing, and we didn't know it was here, and it's made inside the cells, and yeah, whatever. Deal with that. Yeah, basically. We don't know exactly what it does from this, but yeah, it's a thing that's there and a thing that we need to pay attention to. So that's an extra gene that SARS coronavirus 2 probably makes and may or may not have some function on the actual disease. So cool. Next one, and we got 2 left, I think. So this one's looking at G quadruplexes, which is a little pattern where you have 2 or more G's in a row and they're spaced out, and you got to have 4 of these little bunches of G's within a certain amount of space, and then you can say that reasonably, reliably, these are going to fold up into a special little shape where they kind of twist around each other like this. These 4 sets of G's are going to twist up and lock and make a particular shape. This particular shape turns out to be important. It's a thing that viruses can recognize. It's a thing that just is recognized by proteins. And shown here, they've actually modeled the SARS coronavirus spike protein and what they call these G tetrads, structures, or G quadruplex structures, because there's 4 sets of G nucleotides together. And there is a nice little binding pocket right on the side of the coronavirus helicase, NSP13, that seems to bind these. And so it's saying that the reason why helicase may be so conserved may be that it is reading these out. And this tells it, like Kenny Rogers the Gambler, when to hold them and when to fold them and, you know, yeah, whatever, yeah, when to do its job, I guess. So this is a theoretical paper. They do identify these things. They do prove that some of these structures fold up into G quadruplexes. It's a hot, spicy hot area right now, and so this is what all the cool kids are talking about. So yeah, now we can all talk about it too. And the last paper here is a crystal structure between NSP10 and NSP16. So 16 is a methyltransferase that is involved in the camouflage of the virus RNA. It makes the virus RNA look like a cellular RNA. NSP10 is this cool little key protein, and it sticks to NSP16, which is a methyltransferase, and it sticks to NSP14, which is the other viral methyltransferase, and it turns both of them on. It seems to only stick to one at a time, at least as far as I've seen, because 14 and 16, those 2 parts of them, I believe, are similar looking domains. But yeah, so there we go, and here's the structure. And so your little 10 is at the bottom with its 2 little zinc molecules in there. Those help it fold up and help it to stick, at least this one does. I'm not sure what this end sticks to, actually. I should go look that up and see if it's known, but that seems like a little arm that's coming off that's going to stick to some other, probably viral or maybe cellular, but probably viral protein. And it'll just help get this enzyme into the right place to do the job that it needs to do. And then, yeah, they're showing you on there where is the basic patch, which is a nice little long line. And so that's probably where the RNA is going to sit as it is being methylated, and your active site is right here. And so it's going to be, yeah, slide right on here, get locked into place, and then bam, get methylated. Get yourself methylated. So, yeah, there we go. That's our little structure of those. And we had those structures for SARS coronavirus and I think feline coronavirus, but now we have them for SARS coronavirus too. So, yeah, take that, SARS coronavirus. We know something else about you. Here's the paper. That's the authors and what they did. And that is our super long, but hopefully somewhat useful roundup of, what was it? Yeah, 16 papers, a lot of papers. Thanks very much. This has been Ben Newman.